Then I would start the webinar now. I started the recording.
Okay, thank you. Hello, everyone. Welcome to the day 2 of Target 2035 kickoff webinar series. My name is Milka Kostic, and I'm the Chemical Biology Program Director at the Dana-Farber Cancer Institute, which is a training and teaching hospital of Harvard Medical School in Boston, Massachusetts. And I will be your moderator for today's session. So in yesterday's session, we heard from speakers and panelists about the reasons behind Target 2035. We heard the motivation, the why. Today's session will focus on the what. What targets should Target 2035 focus on and how to prioritize them and why. So to help us think about the what question, we decided to split today's webinar into 2 one-hour sessions. The first session will focus on examples from the recent past, while the second session will take us into new target areas. And just to explain the logistics of the 2 sessions, each session will feature 3 15-minute talks, followed by question and answer and discussion with audience. So we ask you to type in your questions using a question and answer function in the Zoom, and you should feel free to do this at any point. We'll go back to those questions after the first 3 talks, and then we will continue after about 10 to 12-minute discussion into the second session. Please note that you can upvote the questions, and by upvoting, you can signal your interest in a specific question that should be prioritized, for example, is more interesting, relevant, whatever your view is. Also note that these webinars are being recorded, and the recordings will be available on the Target 2035 website by the end of this week. So in case you missed yesterday's session, or you need a refresher, you can always refer to those recorded webinars later. Last point, there is another webinar happening tomorrow that will go into the how question and talk about some of strategies that we can use to accelerate Target 2035 efforts. And there is another webinar on November 16th that will happen in a time slot better aligned to those in Asia Pacific areas. So stay tuned for that as well. At this point, without any further ado, let me introduce our first speaker. Our first speaker in this session is Stefan Knapp. Stefan is the head of Structural Genomics Consortium site at Frankfurt University, as well as professor for pharmaceutical chemistry in the Institute of Pharmaceutical Chemistry. He is interested in the development and rational design of selective inhibitors targeting key signaling molecules. And I believe that today he will share his experience in targeting kinases. So Stefan, I will hand it over to you.
Thanks, Milka. So I hope everybody can see my screen. And so I would like to thank the organizing committee giving me the chance to talk here and present some of our work. So I named my talk expanding the scope of kinase chemical probe development. And I think it's an important aspect that how we target kinases, and I will go a little bit more into detail of structural features of different inhibitors and what they do in cells. So first of all, we start off, we reviewed the kinase area now a decade ago in 2010, and what we observed, and I think this is now very well known, is this bias that we have towards a few studied targets. And here you see for the kinases that's particularly severe. So we study very well about 5 to 10% of the kinases, and the rest is less well studied. And just updated the papers in PubMed. So we are now close to 1 million papers. So there's an enormous knowledge in kinases, but it's very, very focused on a few targets. And that has now been shown for many protein families. It's not kinase specific. And it's probably due to many reasons we have how we do research. So since then, since this 2010 review, a number of inhibitors that we highlighted during that time in clinical trials indeed made it to be approved, among them the CDK inhibitors, the JAK inhibitors. All this has happened the last 10 years. And interestingly, there are also new kinase targets still discovered, and since the 2001 kinome paper from Manik, about 30, 40 new kinases that have been discovered still. Now, in 2020, I think the situation is still pretty much the same. Here, the number of papers, but at least the awareness of the problem is much more pronounced. So now we have the NIH IDG program. Our colleagues at the UNC assembled the first set targeting the kinome called KCGS set. And down here you see the coverage of inhibitors for these kinases. It covers about 50%. And we just got funded to continue this work together with the UNC to cover the entire kinome with narrow specificity inhibitors that we call chemogenomic compounds. So first of all, for those who do not look at kinases structural biology every day, I just put a very simple introductory slide of a kinase, and you see the bilobal structure that is conserved in the entire family, the upper and the lower lobe. The ATP here is squeezed between these two lobes, and phosphate transfer happens here on the surface. You see this a little bit clearer here, substrate peptide bound to a kinase, and here's the ATP binding site. So we usually target the ATP binding site, and if we target the active state of the kinase, you call this a type 1 inhibitor. If we target an inactive state that is characterized by this tripeptide motif DFG, I talk about that in a minute. At the back of the pocket, you call this a type 2 inhibitor. Then we have allosteric inhibitors of various kinds that we call type 3 and type 4. And interestingly, these inhibitors behave very differently in the cells. So the kinases have a high degree of plasticity. And this plasticity, of course, enables us to target different pockets in this kinase for the development of modulators. And so we can target not only catalytic activity, but also a very important scaffolding role kinases have in cellular signaling. First of all, it's one of my favorite movies, just shows the transition of a DFG in and out. You see how dramatic the structural changes are. This is based on a few crystal structures with a tethered molecular mechanics simulation that opens here this pocket. And if you look at the effect on the binding site, it is very significant. Here we have the DFG in conformation and the active state of the kinase. And here you see how the pocket is extended in this DFG out, DFG motifs located here, that allows us to target this back pocket. Then furthermore, if you look in the PDB, the structure database, you find a lot of different targeting pockets in kinases. Here I highlighted some. They are clustered all over the catalytic site and have been explored to various levels. There are some approved drugs for the targeting the kinase MEK, characterized by really high selectivity. Is the inhibitor that is currently marketed, and they target just a back pocket in a non-ATP competitive way. So if we look now on a cellular level, what happens when we target such kinases, we see that the binding mode of kinases influences the cellular consequences on signaling. Here one example, an inhibitor that we called type 2B because it doesn't target the DFG, it targets here this alpha C out pocket, so just opposite this DFG pocket, and that distorts the kinase. So the kinase is no longer recognized as a substrate, and also the downstream signaling is abrogated. You see this quite nicely in these Western plots. You see here a robust effect in downstream signaling. This is a washout experiment. So we wash the inhibitor out, and then we look at the recovery after 1 or 2 hours. You see about 2 hours, we have the signaling back of this kinase called LRRK. But you also see the kinase does not get activated by upstream signaling partners. So also we abrogate activation as well as downstream signaling. If we look at a type 1 inhibitor instead, you see there's no effect on the activation of the kinase. In the contrary, it's in an active conformation, so it's easily phosphorylated. And of course, we have also effect on downstream signaling. That means this mode of inhibitor binding modulates the way how we change the phosphorylation network in cells. Sometimes we even see activation if you incubate kinases with inhibitors. That happens, for instance, here in the case of AKT, a very important target in oncology. If you use a type 1 inhibitor, we see, of course, very robust effects on downstream signaling. But if you look at the activation of AKT, we see that AKT is strongly activated in the presence of this type 1 inhibitor. And that is because we stabilize the active state with the inhibitor, and AKT is recruited to the membrane where it's activated. We see this very nicely when we compare the lipid or membrane interaction of AKT, and you see that you get here strong signals for this phosphoinositol phosphates if we incubate the cells with a type 1 inhibitor. The type 2 inhibitor doesn't do this. You see AKT is not activated. And we see also that it's not getting recruited to the membrane. So also here, we will have very different effects in cellular signaling depending on how we target the kinase. So in essence, that means for this large family, we not only need to make ATP mimetic type 1 inhibitors, it would be really neat to have inhibitors that allosterically target the kinase, so we can dissect signaling networks based on their allosteric function as well as on the catalytic function of the kinase. Here's one example that we just published, a kinase called LRRK2. You see on the left side, it has many cellular functions regulating autophagy, endocytosis, cytoskeletal functions, and so forth. And this kinase has been now extensively studied, mainly because of its role in Parkinson's disease. So 9 of the missense mutations in LRRK are sufficient to cause Parkinson's disease, and it's one of the main risk areas of inherited as well as sporadic targeting disease. It plays also a role in a number of other inflammation processes, and there's still a lot to learn about this kinase. We as a structural biology lab that is mainly focused on crystallography try to crystallize this kinase, which is huge, it's 2,500 residues. And we got crystals, and we have been playing for years around with these crystals, but the diffraction limit was always around 7 angstrom. Then we started collaborating with Andres Leschinger group at the University of California in San Diego. And in a few weeks, I must say, got a very nice about 3 angstrom EM model, which is shown here, that has all the catalytic function combined of LRRK, which contains a small GDP binding domain, an intermediate so-called core domain, a kinase domain, and a WD40. Also all the Parkinson's disease mutations, also mutations of finding Crohn's disease mapped to this area. So LRRK is, has a typical kinase fold. You may recognize it here in this 3D image of the kinase, but apart from its catalytic function, it's mainly phosphorylating RAP GTPases. It also has a scaffolding function. And one of the known functions of LRRK is that it binds to tubulin. This is a 14 angstrom tomogram of tubulin structure binding LRRK kinase that is wrapped around it. And by doing so, of course, we influence cellular transport. You may remember all cellular transport is mediated by 2 families of proteins. One is called the kinesins that walk from the minus to the plus end of the microtubules. And the dyneins, they do transport in the other direction. We can monitor this by immobilizing tubulin here on a glass support and look at the transport. And if you incubate here the tubulin with the part that we crystallize and also EM structure determine, you see that you get a strong inhibition of motility along the tubulins. So coming back to inhibitor development, what we observe is quite interesting. So if you take type 2 inhibitors, and I showed you they bind the allosteric back pocket, we abrogate recruitment of LRRK to tubulin, whereas if we take type 1 inhibitors, here are 2 examples, we strongly induce tubulin binding. And then again, showing that we will have here an effect that comes from a scaffolding function that we induce based on the inhibitor type. Quickly, in the last few minutes, just an example of an allosteric inhibitor that we developed targeting the LIMK kinase. And this inhibitor is important for modulating also cytoskeletal function by phosphorylating cofilins. It's downstream of small GTPases of the RAC-RO family and is regulated by ROC and PAC kinases. There are many reasons targeting LIMK kinases. Here are some of them listed. We were particularly interested in fragile X chromosome, which carries a unique isoform and leading to the activation of particular linkase 1. We developed inhibitors based on a published fragment structure that was published a few years ago. You see how the inhibitors bind and the changes they induce when they bind into this back pocket. And we have here with this inhibitor possibility to reach the DFG out pocket. You see this pocket here, the ATP side as well as the alpha C out pocket. So the inhibitor binds here in the back pocket, not where ATP binds. This inhibitor is highly selective. You see here the selectivity profile of the inhibitor, just targeting LIMK 1 and 2, screened against the kinome. And we validated the inhibitor by different in vitro techniques, determined KD by ITC, in rapid fire MS, that means direct phosphorylation of peptides, as well as in Brett assays, assay format that we use in cells that uses a tracer thread pair with the full-length kinase. So phenotypically, if we use these inhibitors, LIMK is not recognized by PAC, which is the upstream target, because we distort the kinase. And we also see more significant phenotypic effects. I just brought you two pieces of data here. One is a MS study where we look at the phosphoproteomics and the most significant changes that we observe in LIMK kinase phosphorylation when we add a type 1 inhibitor that I haven't presented here are not conserved when we use the type 3 inhibitor. The changes are shown here. And that leads to different types or different efficacy in inhibiting nurid outgrowth. A phenotype we look for the fragile X chromosome disease. And you see, in particular, the type 2 binding mode seems to be very efficient inhibiting this phenotype. So that opens the possibility targeting now kinases that are completely inactive catalytically. Here we could also target just the scaffolding function. These kinases are called pseudokinases and represent about 10% of the family. So they're about 50 pseudokinases expressed in human. They are associated with many diseases, so they have very important scaffolding functions. They are usually catalytically inert. Some of them have very low catalytic activity. And we can target them either allosterically, and of course we can also develop products, something I haven't talked about. And just recently we released our first inhibitor targeting a pseudokinase called CASC. And you see in this crystal structure how we did that. The unique change in this pseudokinase is that the usually very conserved DFG motif is changed to GFG, and the introduction of this glycine here produces a large extra cavity that we can target and make this inhibitor very selective for this kinase. So with this, I hope I convinced you there are a lot of additional possibilities beyond just making inhibitors for kinases. I think it's an exciting area to work in right now. And I think we need to understand much more how we can target these kinases efficiently in order to find the most efficient mode of action of inhibitors for a certain disease. So with this, I just want to highlight one initiative that we are organizing here in Frankfurt. I'm convinced pharma and many labs have highly characterized, very good compounds available. We started a program that we called Open Science Probes. So these are donated chemical tools from pharmaceutical industry, from academics. We often put additional profiling data and help the compound developer to meet all criteria. And then we distribute them here from our side and put all the data in the database. So if somebody is looking for a tool, he can maybe find it. Currently, we have more than 70 molecules and the negative controls there. Just remains to thank my group and collaborators. The LIMK inhibitors have been synthesized by Thomas and colleagues in the chemistry group here in Frankfurt. The crystallography was done by Sebastian. A lot of the kinase crystallography is done by Apparatus that are highlighted here. Would like to thank Andres, who did the EM structure of LIMK, Sam, who did the assays on the tubulin binding, Elizabeth doing the EM tomography, Bernard, a long-term collaborator working with us on profiling, phosphoproteomics and signaling profiling. Stefan's group, we collaborated on many probe development projects, and my colleagues at Promega, which we collaborate with to develop a kinome-wide platform. With this, I hope I gave you a good introduction into our work, and happy to answer questions later in that session.
Thank you, Stefan. As we are letting our next speaker, Cheryl Arrowsmith, start her slide shares, let me remind everybody that you can enter your questions using the question and answer box, and we'll come to them after the first three speakers have completed their presentations. So with that said, Cheryl is the chief scientist of the Toronto node of the Structural Genomics Consortium. She leads the SGC program to develop chemical probes to chromatin regulators for target validation. And we will let Cheryl take it over and let us what they've learned over the last 10 years.
Hello, everyone. Yeah, it's a pleasure to tell you about our chemical probe program targeting epigenetics. So just a quick background on what epigenetics is. Start my video, okay. Everybody see me now? Okay. So epigenetics, it relates to how the DNA in each cell is packaged and organized so that you have so you have a single genome in all the cells, but specific expression patterns determined by the packaging of the genome in different cell types. So one genome, many different cell-specific epigenomes. Now the epigenetic changes that determine this are reversible because disease cells also have specific epigenomes. Then the reversal of that has the potential for modulating disease. So there was great interest in this starting in the 2000s because of successes of drugs like azacitidine and in the work in the 90s and early 2000s on histone deacetylase inhibitors. So these HDACs, for example, remove acetyl on the histone tails of nucleosomes on chromatin, opening up or closing up chromatin, and change gene expression. And azacitidine reverses and hypomethylates DNA, undoing gene repression. So about 10 years ago, a little over 10 years ago, with the increasing awareness of the role of epigenetics in disease and the clinical success of HDAC inhibitors and azacitidine, there was great excitement, and even to the extent that there was papers and articles in the popular press like Time magazine, and in the drug discovery industry and in academia as well, great excitement about the potential of epigenetics. And this was fueled by the elucidation from the human genome sequencing and biologists around the world that there are these protein families that we refer to as readers, writers, and erasers of post-translational modifications on DNA, RNA, histone proteins, and many proteins, but much of the epigenetic regulation is at the histone protein level. And taken together, there's anywhere from 300 to 500 proteins involved in this. So we in the SGC decided to target these protein families that read, write, and erase acetylation of lysine and methylation of lysine and arginine. And I'm going to limit my talk to the two largest families that amongst these that we've tackled and had the most success with, and that's the bromodomains that recognize acetyl-lysine and target proteins to acetylated sites in chromatin, and methyl-lysine readers and especially the enzymes that deposit methyl on lysines and arginines. And one of the reasons is that we discovered that these the protein pockets that recognize and also recognize the modifications that bind to them, and also part of the enzymes that modulate these, write and erase these marks, are aromatic rich pockets used for recognition of lysine and arginine, and some structural information is shown here. We didn't focus on HDACs because a lot had already been done, and there were already clinical programs there. So the upshot of 10 years of work on these target families that are largely, and there was no chemical matter for most of them when we started about 10 years ago. The results are shown here with the chemical probes and the protein families. And many of these these are not drugs, but they are chemical probes, potent selective cell active modulators of a protein or a small set of proteins. And the yellow ones circled are the protein targets, where there are also either late preclinical or clinical programs in some of these proteins. And so that use of the chemical probes can be used to elucidate biology, which then can inform on disease states, clinical progression, and also identifying new targets in here that may be useful for pursuing translational studies of. And excitingly, last year, the first inhibitors of EZH2, pyrrolidine repressive complex 2, where tazemetostat was approved for clinical use. So, and the two family papers that we published are shown here on the toolbox for bromodomain signaling and the toolbox for methyltransferase signaling. So what did we learn from this 10-year program of working on unprecedented targets where we didn't have any chemical matter or not a lot of knowledge to start with? So as I promoted yesterday, the protein family approach is highly, highly enabling. It allowed us to build up knowledge, know-how, and expertise, including ability to make the proteins, their three-dimensional structure, how to assay them, reagents, identifying which antibodies are best to use to monitor the methyl or acetyl marks that they make, cell assay formats that work and those that don't work. And if we had just done, cherry picking the one or two most interesting proteins amongst the individual families, I think we would not have gone as far as taking this concentrated protein family approach. And selectivity within the family is very important. By having all the proteins in the family, we were able to assay that. And in certain cases, bromodomains, for example, one can lead hop from chemotypes that are active in one or many members of the family to create selective ones in another member family. And not surprisingly, structural biology was highly enabling for this. Many protein interactions can be drugged. Previous to this, it was thought that protein interactions are undruggable. We've shown extensively that they can be under the right conditions. We also got some surprises, and I'll highlight a few of those along the way. So the bromodomain work was led by Stefan Knapp and his colleagues, first at Oxford and finishing off in Frankfurt. It's summarized here. I won't go into the detail, but just a few take-home messages about this family. Common bioactive chemotypes were identified that had activity across much of the family, and therefore HTS screening and extensive search for new chemotypes was not necessary, but structure-guided optimization of these ligands to make selective bromodomain inhibitors was important, and structure-guided that, and also knowledge of their histone substrates. Well-characterized selectivity in this family was very important because of the very strong phenotype of the inhibitors of the BET family members. And for example, the activity of JQ1 and IBET inhibitors very well demonstrated the importance of the BET inhibitors in modulating gene expression and phenotypes. And if you just even slightly inhibit the BETs through an off-target activity, when you're intending to hit another bromodomain, you may misinterpret your biology. So in Toronto, we focused mostly on methyltransferases and methyl-lysine binders. So protein methyltransferases use S-adenosylmethionine as the cofactor and methyl donor to methylate lysines or arginines. And it was initially thought, by analogy with kinases, that that inhibitors of the or competitors of the SAM binding site could be a great way to target the entire family. But it turns out that this is not so much the case, although we do have some examples shown here. In general, the SAM binding site is more polar, and therefore compounds that bind there tend to be polar and not cell permeable. But more often we found substrate competitive inhibitors that bound in the substrate binding site, as shown for the targets here. We also identified a few allosteric inhibitors amongst these dimeric arginine methyltransferases. And very interestingly, in this subclass here, there are these enzymes, EZH2 polycomb repressive complex and the MLLs that are part of trithorax complexes, we were able to identify allosteric regulators by antagonizing components of their multi-protein complexes. And then finally, some of these enzymes, we were never able to get direct catalytic inhibitors, for example, the NST2s, but we were able to identify antagonists of their non-catalytic domains, reader domains, and these could now be used in the future to create degraders, for example. So what did we learn with respect to the methyltransferases? Chemical inhibition, unlike the bromodomain family, this family is druggable, but we could not identify a pan-family chemotype. So every target was brand new. We had to do lots of screening. The hit rate was low, but when we did identify inhibitory compounds, they tended to be very selective. And so this is shown here by the great selectivity of this chemical probe panel with respect to the enzymes. These are flexible enzymes, not amenable to docking and BLS, and many require protein partners or other domains within the larger protein for activity. Another thing that we discovered in working on the family is that in cellular assays, the effects are slow. It takes days for the biochemical changes to occur. This is illustrated here by day 4 for G9A. Methyltransferase inhibitor is most effective after 4 days of waiting. And then then there's an even further delay before you get phenotypic changes because you have to wait for the gene expression change to occur. And but the good thing is that the methylation marks on histones are good biomarkers of response, as shown here. The increasing dose after 7 days of treatment, for example, you can see this mark go away. So all of those were things that we learned by working on the family together. Now, a surprise we got that we had not appreciated in the beginning, and which is now being used, we take into account when we think about trying to drug methyltransferases, that in many cases, the substrate competitive inhibitors actually nestle right up next to the cofactor, SAM here, shown in white and purplish. These probes are often cofactor dependent when they bind, as shown here. And so from the 3D structure, we take into account where the cofactor is bound, and in doing our assays, we want to have the cofactor bound. Another learning from the family. And finally, I'll just update on the concept of these auxiliary domains. Often methyl-lysine reader domains within these enzymes and other chromatin regulatory complexes. And the domains are listed here, and then estimates of their druggability from Kemble. And in collaboration with Stephen Fry and Lindsay James at UNC, we were the first to identify some antagonists of these types of domains. More recently, the NSD2 and 3 PWWP domains. Interestingly, these WDR5 beta propeller proteins that are subunits of several chromatin regulatory complexes, particularly polycomb complex and the trithorax complex shown here. And this is just an example of a surprise. We and others found that when you antagonize with a small molecule the binding of this stimulatory peptide to the EED subunit of polycomb repressive complex 2, you prevent its catalytic activity as well, or perhaps even better, than direct inhibitors of the catalytic domain, as shown here. So this inspired us to now, in the future, to embark on tackling this entire WD40 domain. These are protein-protein interaction domains, subsets of which are in chromatin regulators, but also in many larger E3 multi-protein complexes. And interestingly, this protein family has the largest proportion of genes that are essential in cancer compared to many other protein families. So we're excited to embark on this as well. I'll finally end with uses of these, of the chemical probes. We like to use the entire collection against patient-derived cancer models, as illustrated here by work with Patty Shakameter, recently accepted, identifying and characterizing how PRMT5 inhibitors are active in a subset of glioma patient-derived models. And that's one example of many of the uses of the probes over the years. And this is compiled by Matthew Shapira of notable publications using these probes, enabled by the rapid sharing of unencumbered chemical tools. We have a click MTA that's easy to, one page to read, one click to access these compounds, and most are commercially available eventually. So we gave out thousands of samples. Many people have discovered new biology, new therapeutic hypotheses, and we think this work, we know this work in many cases has catalyzed new drug discovery programs in some clinical trials. So I will end by thanking my colleagues in Toronto, many of which are shown here. I apologize if there's a few more recent people not shown. Our great colleagues in Oxford and Frankfurt, many of my colleagues here in Toronto, SGC funders, and I want to also shout out to all our chemistry colleagues in pharma and academia who have done a lot of the medicinal chemistry to generate these chemical probes. So thank you.
Thanks, Cheryl. This was just breathtaking to see where things have gone over the last decade. So thank you. We'll move on to the next speaker in this session, that's Brian Roth, who is the Michael Hooker Distinguished Professor of Pharmacology at the University of North Carolina, Chapel Hill School of Medicine. And Brian will take us into a different target space, the G protein coupled receptors, the GPCRs. In addition to GPCRs, his lab also works on ion channels, but today the focus of his talk will be on understudied GPCRs. Brian, please.
Can you see this?
Yes.
And you can hear me. Great. Okay. So thank you very much for this kind opportunity. What I'm going to do is give you an update on this, this program, which is now in, I guess, year 6. The first 3 years were a pilot phase for technology development, and the next 6 years, so we're about halfway through that, is for probe discovery. And this is the Illuminating Druggable Genome Initiative by the NIH. And participating are members of my lab, the Brian Choquette lab at UCSF, John Jin's lab at Mount Sinai. And we focus on G protein coupled receptors. Of course, there's been an explosion of structural information about GPCRs fueled by crystallography and more recently cryo-EM. And this is, for example, a recent structure we published a couple of weeks ago on a interesting GPCR. And one of the nice things about GPCRs, so the receptor here is shown in red, is that there are pockets for small molecules virtually all over the receptor, basically here, which is a common orthosteric site, here, here, and here, which are allosteric sites, and all of those can be targeted for probe discovery. Just a brief introduction of GPCRs. They couple to multiple signaling pathways, these include both G protein dependent signaling, which is shown here for a particular subtype of G protein, G alpha Q, as well as following phosphorylation, arrestins can bind, and they can serve as scaffolds for various downstream signaling pathways. And because of the diversity of signaling pathways, both G protein dependent and arrestin dependent, it really facilitates the design of assays and the discovery of probes which can selectively stabilize or destabilize conformations that are essential for distinct signaling pathways. And currently, in the human genome, there are around 900 GPCRs. About 600 of these are olfactory receptors and are not the subject of this particular initiative. There are around 300 druggable GPCRs in the human genome. And recently, as in last year, I did an analysis of the druggable G protein coupled receptors in the human genome, I guess around 393, and annotated them. So this is a similar annotation that had been done for kinases in the past for compounds that are in the chemical literature. And for this, we used Kimball as a source, as well as structures, which are shown in red. And what you can see here is that for many GPCRs, there are many available compounds, although I must say very few actually selective compounds. And the structural information is fairly well clustered around those GPCRs for which we have chemical matter. And for about half of them or so, there are very few ligands available, almost no selective ligands, and very little structural information, although that is improving as we go forward. So the opportunity that we took on was to identify these understudied G protein coupled receptors, devise scalable assays for them, and then to use those to identify small molecule probes. And our wish list basically is to have an agonist and an antagonist for each GPCR with a structure, that these compounds should be highly selective within the family and across the GPCRome, and that they should be useful for both in vivo and in vitro studies. And the approach basically has been to develop scalable assays for the unbiased screening of the druggable GPCRome. And one of the things that we have strived to do is to make the technologies available open source without encumbrance through Adgene. And to date, more than 25,000 orders have been shipped from Adgene. So this has been really remarkable pickup of the technology. We then combine the scalable assays with ultra-large scale computational approaches to ultimately with medicinal chemistry to identify probes. All probes are then made commercially available without encumbrances of intellectual property. And going forward, we have increasing dependence upon GPCR structural information via cryo-EM. So just to give you a sense of how this is done and how we've been doing it for the last 2 or 3 years, I'll describe some scalable assays that we've made available for unbiased screening of the druggable GPCRome. And the first assay platform that we used relies on the propensity of GPCRs, once they're activated by an agonist, to catalyze the interaction of the receptor with arrestin proteins. And there was an assay that was developed by Richard Axel's lab, Gilead Barnea, in 2008, where a portion of the vasopressin receptor, which has a very high affinity for, when it's phosphorylated for arrestins, was tagged to the receptor with a cleavage site and a transcription factor, so that when the receptor is activated by ligand, arrestin is translocated, the arrestin has a TEV protease, that releases a transcription factor, TTA, which then leads to luciferase. So these are basically luciferase-based screens. And we had previously used this assay sort of in a one receptor by itself role, and it works very well. It's quite robust and scalable. And so what we did was then to engineer this assay through synthetic DNA technology for 320 of the druggable GPCRs in the human genome. And we made this available now about 5 years ago. The resource is called the Presto Tango resource. And this is the tree, the GPCR tree. And wherever there is a colored circle, we have an assay that we have developed using this resource. And you can see that we basically cover nearly all of the druggable GPCRome, with the exception of the frizzled and tastant receptors and a few adhesion receptors. And these basically require other assay platforms. But basically what this allows us to do then is to use this platform in a highly parallel fashion to discover small molecules that modulate the activity. And then once probes are found, it allows us to assess the selectivity across essentially the entire GPCRome. We combine this with ultra-large scale computational approaches. And to give you a sense of how this is done, so there has really been a renaissance in GPCR structural biology over the last 10 years, which is accelerating with the utility of cryo-EM for getting complexes. And a couple of years ago, we set out to basically solve the structures of dopamine receptors. At the time we set out to do this, there was a single dopamine receptor that was known for the structure of the D3. We solved basically the D2 and the D4. And the reason for this was that even though dopamine receptors have been studied to a great extent, there are nearly 100,000 papers on dopamine receptors, there actually are no selective ligands for D2 or D3, and very few for D4. So the ligands that are available and are used commonly have a great deal of cross-reactivity. And we noticed, based on the high resolution structure of the D4, this was a 1.9 angstrom structure, that there was this little pocket here in the D4 that could be targeted potentially by ultra-large scale computational approaches. So my lab teamed up with Brian Choiquette and John Irwin's lab to use a technology we're now calling ultra-large scale docking, or ultra-LSD, and this was published last year. And the approach basically was to take advantage of huge libraries of chemical matter that are becoming available, which include sort of the known chemical matter, as well as small molecules that in theory could be made from simple building blocks. So these are virtual libraries. And we use the Zinc database, which was developed by Irwin and Choiquette. And when this project was done, the Zinc database was a couple 100 million compounds. And we could bin out around 138 million compounds that had good drug-like property were cations for ultra large scale docking. And because of the way docking is done, these were docked in, each compound was docked in up to 10 to the 6 conformations. So this yielded a very large number of conformations. Luckily, they have in hand at UCSF many processors. So the docking experiment per se just took a day and a half, although setting it up took several months. And ultimately, when the compounds were ranked and scored, nearly 550 of them were synthesized and tested against the D4 receptor. And based on the hit rate, we predict that in this library of 138 million compounds, there are at least 400,000 compounds that are selective for the D4, which encompass 71,000 unique chemical scaffolds. And so with a relatively straightforward experiment, we have expanded the universe of selective D4 compounds to such an extent that virtually all the medicinal chemists that work in academia could devote their lives to synthesizing D4 selective compounds. And this is a compound basically that came right out of the screen. This is a picomole potency D4 agonist. You can see it activates G protein signaling better than arrestin, so it's G protein biased. When the selectivity panel was done against the GPCR, I'm using the Presto Tango resource, it was selective for the D4, and binding assays across the PDSP screen also showed it selective. So basically in one step, we were able to identify extremely potent selective D4 agonists. This is the most potent and selective D4 agonist to date. We then decided to tackle receptors for which there were not selective probes, although there are drugs targeting them that are approved. And for this, we took advantage of structures of the melatonin 1 and melatonin 2 receptors, which were recently published by a team led by Vadim Cherezov at USC, in which my lab and many people in my lab cooperated. And this was an effort headed up by Reed Stein from Brian Choiket's lab and Hai-Jin from my lab. And here, 150 million compounds were docked. And out of the docking alone, picomolar to nanomolar selective compounds were identified. These were then optimized by iterative analog by catalog. So there was no directed medicinal chemistry that was done by a chemist that was under our supervision, but they were done at Enamine. And ultimately, they were tested for favorable drug-like properties. And a subset of those were shown to have on-target modulation of circadian biology. And so these were really the first selective melatonin 1 and melatonin 2 agonists and inverse agonists. And they had very interesting effects on circadian biology, which were the on-target activity were verified by studies in melatonin 1 and melatonin 2 knockout mice. So the notable probe successes we've had in the short time this program has been available, we identified probes which are allosteric modulators for these orphan GPCRs, GPR68 and GPR65, for the orphan GPCR, MRGPRX2, for the D4 dopamine receptor. We identified, in collaboration with the Glorium lab, orphan peptides for orphan GPCRs. We've used these resources to evolve nanobody-based reagents for understudied GPCRs. And as I said, we have identified for the first time highly selective and potent melatonin 1 and melatonin 2 agonists and antagonists. Going forward, I think there will be increasing dependence on GPCR structural information via cryo-EM. And we now can achieve a turnaround time with a new chemotype within 2 to 3 weeks. So this is really accelerating our in-house drug discovery efforts. Of course, there is a fair amount of expertise that's needed on the GPCR end to get complexes, but that expertise is available. I just want to thank the funding agencies that funded this work. Of course, the Illuminating Druggable Genome Initiative. This is a NIH Common Fund initiative, as well as the screening center that my lab has run for the last 22 years, which provides very important counter screening and profiling services, not only for my lab, but for hundreds and hundreds of labs around the world. And with that, I'll end. And we can go to the next talk.
Yeah. Thanks, Brian. So I will ask you, Cheryl, and Stefan to turn your cameras on so that we can go to the set of questions we received. They will be directed at, I think what I'm going to do for the sake of time is pick one directed at each one of you and then move on to the next session. And you can feel free to take a look at the questions and then type in your answers for those that we can't address at the moment. So one question we received in the sort of kinase space is the question about what are type 1 or type 2 inhibitors more useful from a perspective of a drug design? So Stefan, do you have an opinion here, or what's your view?
So yeah, I mean, there's not a certain binding mode that is more or less useful. So of course the initial expectation of type 2 inhibitors was that they would be more selective, but this is not the case. So type 2 inhibitors can be incredibly promiscuous, and that means that most kinases can actually assume this inactive state of the DFG out. So, but in terms of their usefulness as drugs, there is currently, from the more than 60 inhibitors that have been FDA approved until now, there are type 1 inhibitors, type 2 and type 3 inhibitors. So there is no certain trend that would tell us that type 1 or type 2 are more selective. The escape mechanisms in oncology, when you get away from the active state of the kinase, are more complex. So the kinase can more easily escape in resistant mechanisms, and but that also, I think, is just anecdotal. There's no systematic studies on this now.
So actually, rather than directing questions to Cheryl and Brian, we just received a question that I would want perhaps each one of you to offer your response to. And that question is related to the goal of Target 2035, which is to find specific modulators within context for nearly all proteins in the human genome. With the presentations that we have heard so far, it sounds like that we really want and need sets of various types of modulators for each protein. So we may want to have allosteric and orthosteric inhibitors, activators, glues, inhibitors of protein-protein interactions. So there is also, so this is the comment from Ralph Mazicek, that we should look for modulators of other macromolecules like nucleic acids, for example. So I will turn this to you, and perhaps we can start with Brian and then go to Cheryl and go to Stefan to address this. How do we set priorities? What's your view on in terms of how do you prioritize targets and chemical matter so that we get the most out of these efforts? I suppose that's kind of where this comment is getting at.
Yeah, so I agree with I guess I'll say Ralph. I don't know Ralph, but I'll just say Ralph, I agree. And for GPCRs, you know, as I mentioned, what we really need is an activator and a blocker, so an agonist and an antagonist for every GPCR. And the reason is that when we go into biology, there are many situations where a blocker does nothing, but an activator perturbs the system in such a way that we can measure something. And particularly for these understudied targets, there's no way to predict in advance whether a activator or a blocker is what you need to see an effect in your system, sadly. So we, you know, if I had unlimited funds, then I would say, unlimited resources, I would say we need, you know, basically to have selective agonists and antagonists for every druggable GPCR in the genome. We've been prioritized by our funding agency to focus on the understudied GPCRs, and that's our goal. But as you can imagine, getting this for even a single GPCR can be quite resource intensive. So what we've decided to do, and I think SGC has basically taken the same approach, is to aim for high quality probes, so quality over quantity, with the idea that even if we only get 10 to 20 probes, people can be assured that these will be useful. We'll not have 100 off targets and that sort of thing. So that's been our approach. But in terms of these other proteins and protein families, I agree 100%. I think there should be a much larger initiative focusing on all the protein families in the genome. We need chemical tools for all of them, but I guess you have to start somewhere. So.
Yeah. And Cheryl, what are your views on
I'll just follow up quickly. Of course, Ralph is correct. We need all these types of modulators, but again, I think the purpose of Target 2035 is to tackle the dark proteome targets where there's nothing, right? So if there's nothing you can do with it, that's where you're going to have the biggest bang for your buck, I would say, in terms of investment and opening up new areas of biology and potential therapeutics. And then once you have that, you've made the protein, you've got some modulator, you create a little bit of knowledge around that target, then it's much easier to do the other things, and I think it will follow. I think the additional types of modulators will follow. And also, and probably as a side a spin out, some of these other targets such as nucleic acids, carbohydrates, et cetera, will come along as well.
And Stefan, what are your thoughts here?
Yeah, I mean, that's pretty much in line with what Brian and Cheryl said. So we were just thinking about this when we wrote the application for our IMI UB Open funding. So what we did there, we split it up in two major topics. One is to look at what we currently work on have and define the best tools, because a lot of people work with that. And I think Brian probably also sees this, that a lot of investigators use 1980 drugs on very well-studied targets, but they're not necessarily the best one. So I think with this, we can make a big impact on what a lot of people do at the moment. And then the other half of the grant is to do something really new. And for this, we're looking at E3 ligases, SLCs, also understudied kinases and other enzymes to build in this new area. So I think we need to find that balance each time we write a larger application. And of course, to do something truly new is much more resource intensive than trying to look at already very established systems. But I think we have to work on both ends to get a good coverage.
Thank you, all three of you. We'll move on to our session 2 now. And the first speaker in session 2 is Peter Sorger. Peter Sorger is Otto Kreher Professor of Systems Biology at Harvard Medical School and the head of Harvard Program in Therapeutic Sciences, as well as the director of its Laboratory of Systems Pharmacology. So I will hand it over to Peter now.
Thank you very much for that. I'm going to continue our discussion of kinases and what we've learned from kinase panels. Even though my session is opening up new target areas, I'm also going to look at what has happened in the past, following up on what you've heard from Stefan in the first section. And in addition, this project is also part of the Illuminating Druggable Genome project that was alluded to by Brian Roth, and the piece that we've been looking at has to do with kinases. So the kinase family, actually, we think about it, it's been around for many, many years, defined almost 40 years ago in terms of some of the activities against peptides. But one of the things that's emerged from the dark kinome project is that it's not actually as trivial to define as you might imagine. And there are a series of overlapping criteria. One of them is obviously structural. So to what extent is there a similar fold? Catalytic activity is important, but the kinases, the family of protein kinases, is extremely similar in many respects, including to the extent to which it's inhibited by small molecule drugs to the lipid kinases. And those kinases, as we all know, the PI3 kinases, et cetera, tend to function in the same signaling pathway. So it's conventional to include those kinases. And then there are a series of atypical and in some cases metabolic kinases that live into that family as well. And if you look back at the historical definitions arising initially from Manning, there's KinHub resource, Uniprot, and then what the NIH has dug together and what I'll describe in a moment as the dark kinome, we find a series of sort of overlapping and non-identical definitions. And so this would be the first step in sort of defining activity in this space, is to get a good feel for what these targets are. So in a re-annotated version that are based primarily on sequence similarity and then on HMM models at the primary sequence level, we've come up with two definitions that might be interesting to people. The first of them is what we are calling the total number of kinase domains, and those include both atypical and what Ralph mentioned, critically also the pseudokinases, which are thought not to have conventional catalytic activity, but nonetheless have function. And historically, a series of enzymes have been left out of this for no obvious reason, and these now get added back in. And this has been one of the products of the dark kinome activity. And then the second one is to create a curated kinase. So this would be a minimum set most similar to the Manning set that we've historically worked with. And this adds about 22 kinases based on structural, functional, or sequence similarity to that family. So for those of you active in this space, I would commend some of this to your attention. And we've now built, as part of the dark kinase knowledge base that I'll describe, a new sort of kinome.org tool. This will eventually be integrated with other tools at SGD and NIH. And this just simply allows you to go through and find these domains based on all of the available criteria, the compounds that bind them, the available knowledge, whether they're pseudokinases, and the sort of half a dozen non-identical and partially overlapping classifications. So we now have a sort of a somewhat updated sense of what this target family is. And the question that we wanted to address is, what is really known about this, updating the paper that Stefan showed you from some time ago. And this derives from a project in our lab which uses machines to actually build out executable biochemical models of protein activity. And this is done both with reading and then what we'll call, and I'll show you this in a second, executable knowledge assembly. And so we have now, through a DARPA project related actually to the one that Brian mentioned, the capacity to read all available pathway databases, the vast majority of the literature, certainly all abstracts and all full text articles that are in the public domain, and to input expert opinion. And this is then read into an executable format that you can display as a database, but you could also build, for example, differential equation models if you want. And this allows us to go through and actually look at the totality of information that's available on kinases, both in terms of their function, their genetic associations, and their annotation. Now, one of the things that we found when actually doing this for this highly drugged class of proteins is that there's a substantial challenge actually in assembling the underlying knowledge. And here I'm analogizing this to genome assembly. And so if you actually use machines to do this, they make some sort of obvious and slightly crazy mistakes. So MEC also will pull out methyl ethyl ketone. That's something you can teach a machine not to do. But what a machine has difficulty with initially is about a quarter of all the phosphorylation sites in the published literature are incorrect by a couple of residues. And that's often because the mouse protein is used for the human, for example, or the start codon's annotation has changed over time in the standard reference database. And people actually will put different fragments of this information, and I've given you a couple of examples here. And so what Indra tries to do is to eventually then assemble this into a single executable comment. And one of the aspects of this we found is really the scope of of what I would call the unknown knowns in this space. So because there has not been a stable naming convention for this protein family, and that extends to all other protein families, but is worse in the case of those that had more historical work on them, it's actually hard to find much of the literature on many of these kinases. And the large-scale databases, for example, the Cancer Genome Atlas, have also not been systematically mined to find recurrent mutations in kinases, which are not otherwise among that subset that Stefan noticed are the most heavily curated and studied. So here's sort of what you can get out of this. There's a version here that you can get for every single kinase. We've now assembled this interaction network here. If you go in, and this is automatically updated, if you go in, it'll actually tell you what a belief score is here, and it has a very precise definition of what each of these edges are, and you can find the underlying literature. And the critical thing is this Indra statement here. This is what a computer can now read and build a much more sophisticated model than you're looking at here. So if you actually go ahead and do that, and again, this sort of recapitulates using sort of machinery rather than human annotation, what is actually known about kinases, and the number of the definition here of statement are either this 10x score that I won't describe, or these Indra statements that I've just told you about. And what you'll see is that basically the amount of annotations spans 10 to the 4th logs, with a lot of the attention and the most selective inhibitors being those highly annotated kinases. And the dark kinome has effectively been defined for the purpose of this investigating druggable genome as roughly the bottom third. But you'll see that it's a continuous distribution. And as we continue to improve the machines, we actually move this graph here to the right on a systematic basis, because we're actually finding new information even for the darkest kinases, and very few of them have nothing on them, but it's often difficult to find and buried away in the literature with non-conventional naming characteristics, and particularly in large-scale databases. So if we were to take then that understudied, under-annotated kinome and project it onto the choral tree, so this is the standard tree for showing that, with, say, tyrosine kinases up on the upper left and the AGC kinases down on the bottom. This is redrawn from a phylogenetic tree based on hidden Markov models, so it's a sequence-based comparison of the catalytic sites. What you'll see is sort of an intermingling of the understudied kinases, which are in blue, and the well-studied kinases, which are in yellow. I've zoomed in on just one little area there of the CDKs, because of course those are famous. And you'll see that there's no clear pattern here. There's sometimes clusters of understudied ones, but in other cases, look at HIP1K here, for example, one of the two isoforms will have been well studied and the other won't, even though when we go into mutational databases, we see that they can be differentially mutated in disease. we've actually done a fair amount of functional characterization that you can find in that bioarchive paper to ask, is there any evidence that these are expressed? In general, the answer is yes, they're not less likely to be expressed than like kinases. And what genetic data is available to show that actually they might be functional from either high throughput CRISPR annotation or from annotation of human variation. And so there's a sort of a good scattering throughout the kinase tree and decent evidence for about 2/3 of them that there's function in at least some cell types. So it makes it an attractive target, but also a challenging set of targets because of the structural diversity. The one area in which, in general, the annotation is the best, and there's been the most chemical development, not surprisingly, are in the tyrosine kinases, in particular, the transmembrane receptor tyrosine kinases. So we've now assembled a version of a drug target pair database here, the small molecule suite, which was published last year and was the thesis project of Ninka Morit. And you can see that online, and you can also find it by a Google search. And the difference between this and other knowledge sources that have been created is this is completely machine driven. So we go through with humans and we recheck that the machines haven't made mistakes, but we don't put expert knowledge in. We actually drive this directly by data. And if you're interested, you can go through and find for any small molecule target, but here's kinases highlighted, the name of the series of compounds, the symbol for the gene, of course, and then what sort of selectivity class we have. And I'm going to, in the interest of time, not go through those selectivity classes, but they approximately correspond to what you might expect, which is the most selective compounds are those with both the highest affinity and the smallest numbers of targets. And then you can take any number of these things and compare them with each other. So it looks like the type of tools that Tim Wilson and his colleagues have put together, but in this case, driven out of available resources at Kemble or that can be accessed by automated machine reading. And the hope then is to create a resource that can be continuously upgraded without human intervention. Now, the question that arises, of course, is how selective are any one of these compounds for a small molecule target? Because we were, like Tim, and SGD in general, very interested in trying to build out optimized sets of existing small molecules, both for the light and the dark kinome. And the type of assay that's used in this space, and now increasingly in others, including in epigenetic regulators and bromodomain enzymes, et cetera, is some sort of a profiling assay. Here's an example that's based on proteomics, in which cells are grown, extract prepared, drug added, the little triangle, and then a bead which contains a series of drug-like, relatively low selectivity drug-like molecules is used to pull down the extract in the presence of the test compound, the triangles, and then this will compete those protein targets off the bead, and in principle allows you then in a mass spec to go through and look at entire families of kinases over a dose response curve. And there are versions of this that are also done using recombinant enzymes. And this allows you to get sort of a family-wide perspective on the selectivity of available compound families. And this has been done fairly extensively by a variety of investigators. And we've done some of this work as well. Now, one of the things we find in almost all of these cases is that these compounds, and I'll show you more about this in a moment, these compounds only very rarely, including clinical grade compounds, only very rarely hit a single target. And so the average number for most selective compounds of relatively high affinity binding is between 5 and 10 kinases. And so this polyselectivity arises from the fact that the binding sites, as was really nicely shown by Stefan, are actually fairly similar to each other, and the ability to get selectivity involves sort of crawling out of the ATP binding pocket either into proximal residues or relatively less commonly into secondary sites, the allosteric sites that are used for MET kinases. This is a case where we've, in collaboration with Jeremy Jenkinson's group at Novartis, gone through and actually clustered all of the kinases on the left based on their inferred fold in the catalytic pocket as judged by small molecule binders, as opposed to by sequence similarity. And you see they fall in this particular clustering into 9 families, which if we then map them back onto the kinase tree, this is actually family number 8, you'll see that proteins with similar structural properties and also properties in terms of binding small molecules actually are scattered across the phylogenetic definition. So this is what we often see in the polypharmacy of these compounds. They'll hit a couple of what we think of as closely related kinases with respect to coral, but then they'll hit a whole bunch of other things all around the tree. And of course, we can now at least begin to rationalize it by these Bayesian affinity fingerprinting type methods, these chemoinformatic methods. We're trying to get much deeper insight into this because at the moment, all of this adventitious polypharmacy is really a consequence of the binding pocket, and it tends to come up after the fact. It's very difficult to design to this. And so here's a machine learning method. Another graduate student in the lab, Chang-Chang Liu, who's working as part of DarkKinome, actually trying to go through and actually improve our ability to do extremely high resolution docking, not using physics-based methods, but now using these machine learning-based methods. And if anybody's interested in that, I'm happy to tell you more about it later. Our goal in this case would be to get to the point where we can actually design the polypharmacy of these molecules rather than simply discover it after the fact. One thing we have already noticed in the DarkKinome project, and this has been delayed in its experimental tests because of the COVID-19 shutdowns, is that there are a fair number of dark kinases, here are a bunch of them listed, so these are among the least studied kinases, in which small molecules that have actually made it into clinical use, these would be phase 4 or currently in phase 3, actually appear to have relatively high affinity binding, and that means these would be in the top 5 targets or so of those compounds. So there may already be important activity against the understudied portions of these kinomes we just might not have recognized. So that brings up sort of a critical question to consider, and that is, what really defines the affinity and the selectivity of any compound? And the challenge is really that we're overwhelmingly influenced by our lack of knowledge of this selectivity. And even for most of the kinases that are currently in the libraries that one buys for these high select kinases, we find that only a subset, typically less than a third of the kinome, has ever been analyzed. So nonetheless, given that information, we can ask ourselves, where are we in evaluating the SGD, IGD type of criteria? So if we said we wanted in vitro biochemical activity, we wanted 30-fold selectivity, using these criteria, and where we try to correct for the absence of knowledge of many of the kinases, we've actually been through and asked ourselves, how likely is it that we're going to actually find these? And we find for kinase inhibitors, other than the allosteric inhibitors, which are hard to assay using these high throughput methods, trametinib, cobimetinib, et cetera, mainly MEK inhibitors, there actually are only 2 extant probes, we believe, that meet this criteria. And in all probes that we can scrape from Kemble, we think there are only 70. And the primary complication here is finding this 30-fold selectivity. So our work has suggested that actually unrecognized polypharmacology is in fact a near universal property of small molecules in this class when you're going at a multi-domain family. And the more we scrape the literature and the more we actually reperform these experiments with more sensitive mass spectrometry methods, the more we find that even what we think of as relatively selective drugs actually have multiple targets. And in work not yet complete with Novartis, but coming out soon, we suggest that in fact this may be therapeutically advantageous. So let me just leave you with that. Kinases are the most extensively targeted protein family, not surprisingly. This is judged based on the number of approved therapeutics, over 150. Defining membership even in this classic family is a little non-trivial, and it's partly because we have different criteria based on phylogeny structure, chemoproteomic features. We're most influenced in the current work by the chemoproteomic features, namely what is bound among known or newly developed ligands. The knowledge of individual domain varies a lot. We heard that also from Ralph. But there's a lot of information that's actually there that, in a sense, we've already done, experiments we've already done that we don't actually currently access. And that's because systematic knowledge assembly is actually surprisingly challenging task. And then finally, just leave you with the notice, with the notion that actually, in some cases, unrecognized polypharmacology is extremely common. And we think that that's a consequence, of course, of the similarity of the binding pockets. It may violate some of the criteria we have, but it may be that that actually is a therapeutically advantageous feature of these kinases. And so much of our work, even in the dark kinome, is now directed not only at finding new probes that would be relatively selective against individual compounds, but actually designing in the polypharmacy with purpose from the first place in order to achieve the kinds of polyselectivities that we think would be advantageous, both from the point of view of probe design, but also from the point of view of eventual therapeutic development. So a fair number of people have contributed to this. I tried to mention some of them along the way, and all of our work on kinases has been done jointly with Nathaniel Gray. Thank you very much.
Thank you, Peter. We will move now to the next speaker in this session. It is my pleasure to introduce Sarah Burlich. Sarah Burlich is a colleague of mine. She's heading the cancer chemical biology at the Dana-Farber Cancer Institute, and her main interests are actually pushing us into a new class of targets, the ubiquitinating enzymes, or DUBs. So I will let Sarah tell us more about their efforts to find chemical matter for DUBs.
Okay, well, thank you, Milka. I'm gonna assume everyone can see my slides since I don't see you shaking your head otherwise. You go ahead and just get my pointer here. But I'd really like to, thank you, Milka, and all the organizers for the invitation. It's really an exciting opportunity for me to be part of this great lineup and, share with you the work that my lab's doing to open up the emerging target class of ubiquitinating enzymes, or, DUBs, as we call them for short. And so for this particular forum today, what I really decided to focus on was talking about the approaches that we've been taking to tackle this new area, really kind of going back 4 to 5 years to think about how we got started when, there was next to nothing known about DUB inhibitors, and, there are no DUB-focused libraries, and then, seeing where that has led us. And so I'll start with just my, kind of 1 minute introduction to the deubiquitinating enzymes, just to be certain everyone's on the same page. These are a family of proteases that regulate the post-translational modification of ubiquitination by cleaving ubiquitin from substrate proteins to rescue these substrates from proteasomal degradation, or to impact their activity through non-degradative mechanisms. The family is comprised of about 100 enzymes, 100 proteases that are split into 7 families. 6 of these 7 families, comprising about 90% of the total DUBs, are cysteine proteases, and then there's 1 family of zinc metalloproteases. We know from structural studies that the cysteine proteases, they all have conserved active sites that comprises a catalytic triad that includes the catalytic cysteine that will cleave the isopeptide bond, linking ubiquitin to its substrate protein, And so, the fact that DUBs have conserved active sites, combined with some other structural features, really gave us hope when we started thinking about DUBs 5 years ago that they would be amenable to target class approaches where we can use the same chemical libraries to really accelerate development of probes for many of the family members, like has been so powerful in the kinase field. So in interest of time today, and given that, understudied enzymes are the focus, I'm not going to, share with you any specific examples of where DUBs are really important in biology, or, convince you that probe development is justified. I'm just going to skip ahead to what is usually, my summary slide on the background of DUBs to say that, it's pretty clear, I think, that based on a combination of literature as well as what's come out from some large-scale genetic, screens, that, DUBs are important in many different aspects of, biology, significant therapeutic relevance. We think about them mostly in the context of cancer, but this is certainly not the only therapeutic area, where DUBs, I think, are candidate drug targets. And where, I hope my lab really can have an impact is in developing the chemical probes that can then be utilized by us and distributed to the community So researchers can really validate and prioritize these dubs and, dig into some detailed mechanistic questions. And so my lab's approach really has been to establish a platform for inhibitor discovery that's target agnostic within the family. And so the process really will initiate with a library versus library screen of a dub-focused small molecule library against a library of dubs to generate a dense matrix of dub-ligand interactions. We then can select some of these dub-ligand interactions of interest and validate them at the level of the purified protein, target class, as well as proteome-wide, which of course then will lead us to a number of interesting avenues. It gives us a number of hits and leads for medicinal chemistry programs to turn these initial hits into high-value probes. We then have an annotated library that we can utilize for phenotypic screens. And then of course, this is an iterative process where with each screen that we do, we learn more design strategies that can go into generation go into informing design of our next generation libraries. And then, once we have rigorously characterized probes, of course, we then can, use these to test and generate hypotheses. So, really the challenge for us on day one when the lab opened was that, we could map out on this slide what we would really love to do, but, we didn't have any of it. We didn't have a single biochemical assay up and running. And really, perhaps the biggest challenge was, we had no idea what a dub-focused small molecule library should look like, or if, one would even be successful in identifying probes for a number of different dubs. So, we had to think about where do we really start? And so we decided to do two things to really get started. The first thing that we did, we said, let's just pick one dub. Our general strategy is, of course, to think of dubs as a target class, but, let's pick one so we really can use it to develop a roadmap for rigorous characterization of probes and to, start to understand maybe the best approaches to studying function of these dubs that, very little is known about many times in the literature. And then while we were doing that in parallel, we really just needed to start doing some high throughput screening. So that we could really generate knowledge for target class focused libraries, both from known inhibitors as well as, from diversity libraries. And so this one dub that we decided to start with was USP7. And although it is one of the most well-studied dubs, the truth is the reason we decided to start with USP7 is because it was the only dub that we could find chemical matter at the time that we really thought had a good chance of being optimized. We identified this hydroxypiperidine-based compound 1 in a patent that had published almost a decade earlier. We were able to confirm that it inhibited USP7, although it was quite weak. It did have nice selectivity. And what was really great for us in this project is we were able to succeed early on in obtaining a co-structure of this compound bound to USP7, which showed us that the compound was binding near the active site catalytic cysteine, really in the channel where the C-terminus of ubiquitin approaches the active site. And then we could observe, a number of key hydrogen bonding interactions and then some other opportunities to maybe improve non-covalent interactions. So our real design strategy was to both improve non-covalent interactions as well as extend this compound to the catalytic cysteine to try to improve potency through making it a covalent inhibitor, as well as really to address this question of, can we have a highly selective compound that targets this invariant catalytic cysteine? So to summarize a lot of chemistry, we were able to really succeed in achieving both of those optimization goals to generate a highly potent and selective covalent USP7 probe compound, XL177A, which is now a sub-nanomolar inhibitor of USP7. And we could pair it with its enantiomer as a negative control. So I think, the two important points just to make here, this was a really important, I think, advance for us because it really gave us confidence that we are going to have the ability to develop good probes for a number of different members of this family. I don't think that's such an open question at this point. You know, work from a number of groups have now shown you can get, you know, high quality probes for dubs, but, you know, that was, wasn't so clear a couple years ago. And then really importantly, through the optimization process, we're able to develop our platform for characterization of dub probes. And so I'll just discuss this very briefly in the context of characterization of the USP7 probe 177A. So we rely on a simple fluorogenic assay for dub biochemistry that measures the ability of the enzyme to simply cleave rhodamine from ubiquitin. This is what we use in USP7 biochemistry. This is the same assay that's used by Ubiquigent, where we do selectivity profiling against a panel of about 40 dubs, which showed in the case of USP7, of this inhibitor, that it was highly selective for USP7. And then, of course, since we're working on a covalent inhibitor here, as well as a number of our other projects, we use mass spectrometry to confirm covalency and labeling of the catalytic system. And then really, I think probably the most powerful kind of dub-specific assay that we use to characterize compounds is activity-based protein profiling. So in these experiments, we'll take lysate, treat with inhibitor, then co-incubate with a dub activity-based probe. These are comprised of ubiquitin that has an electrophile that will irreversibly label the catalytic cysteine of dubs, and then biotin for an affinity purification handle. What we're really measuring is the ability of a compound to block labeling of dubs in lysates by this activity-based probe to infer target engagement and selectivity, which showed, just as we saw with the purified enzymes, that for this compound, it's highly selective for USP7. And then finally, we have no idea what, other targets dub inhibitors may frequently, cross-react with. So whenever we can, look proteome-wide at additional targets of our compounds, which in the case of this particular probe showed a high degree of selectivity for USP7 proteome-wide. And so I flipped through those assays pretty quickly, but hopefully you can appreciate that we're really, well set up now to rigorously characterize inhibitors, new dub inhibitors using a combination of both dub-specific as well as, general characterization platforms. And that in this case, the platform has really qualified our USP7 compounds as, high-value probes. So the next challenge, really for us was, figuring out the smartest way to go about studying dub function when we didn't really have a dominating hypothesis for what an individual dub does. And so for USP7, we've really been able to make progress here by really leveraging what's existing in the literature and from large-scale profiling studies, and then combining this with profiling data that we generate with new inhibitors, which includes, transcriptomics, profiling against large panels of cancer cell lines, as well as doing, global proteomics experiments. And so I just, really fit a lot of data, onto this slide that I don't want to talk about in detail. But, really just want to say that, with all of this data in front of us, there really were two things that, jumped out to us, two strong hypotheses for us to follow up on. The first thing that really kind of jumped off the page was that USP7 is playing a, important role in regulating the MDM2 P53 axis. Which was, not unexpected, as this is MDM2 is a reported substrate of USP7, but it's also reported as a substrate it's also reported to regulate 40 other substrates. So, the fact that it was so dominant was a little bit of a surprise. I won't talk through all the data, but where this was particularly striking was when we profiled the compound against a panel of, 700 cancer cell lines at the Broad. And what we saw was that the profile of our compound was nearly identical to the profile of MDM2 inhibitors, CRISPR knockout of MDM2, and that a mutation in P53 predicted for insensitivity to our compound. A second interesting association we were able to pull out from this data set was between USP7 and PRC, the polycomb repressive complex 1. And so those were, the first two associations that we did a little bit of validation, that we've done some validation work on. I'm not going to go into detail, but just really to summarize that, we've been really pleased to see that the hypotheses that we can generate from, using this existing data as well as, new profiling data has led to hypotheses that really, have panned out. It seems to be a really powerful approach for approaching DUB function. In the case of, the USP7 P53 link, we've been able to confirm across multiple lineages that P53 predicts response to USP7. Here I'm just showing you some specific data in Ewing sarcoma, which is one of the most sensitive lineages. And then I noted that we had an association between USP7 and this polycomb repressive 1 complex. And so through a really exciting collaboration with Kathy Wu and Dave DiCaprio, we've been able to follow up on this and determine that USP7 is eliciting PRC1 dependent increases of MHC class 1 antigens on the surface of Merkel cell carcinoma cells, which then sensitizes them to immune response. And so, there's certainly more to do to, really dig into both of, these particular links as well as USP7 biology more generally. But, I kind of want to stop there in our discussion of USP7, just to really summarize that, this decision to start with one DUB to really develop a roadmap has, worked really well for us. And so we're now really able to apply it to a number of new projects that have been coming out of our screening efforts. I'll talk just in the last, 2 minutes here, about some of these screening efforts and, what they've led to in terms of, target class libraries in our lab. I don't want to talk in detail about high throughput screening and target validation, so I'm really just summarizing a tremendous amount of work here on this slide where we've executed two big screens, one of reported DUB inhibitors times 43 purified DUBs. And then a second in a great collaboration with Novartis Institute for Biomedical Research of a 50,000 compound diversity set times 8 DUBs. And then we've been able to take a number of these DUB ligand interactions and fully validate them. And this is, on one hand, given us a number of starting points for projects focused on individual DUBs, but really towards our, ideas of having a platform in DUB-focused libraries. Importantly, it's enabled us to now design and synthesize our first DUB-focused library, which is really directed at the active site of DUBs. These are covalent compounds designed to target the active site cysteines. We've now synthesized, the first version of this library, which is just over 200 compounds in size, and we've profiled it against a library of just about 60 DUBs, native DUBs, using our competitive activity-based protein profiling platform. Here's just a heat map showing you the results where we were able to see here, darker red indicates the ability of a compound to block labeling of that particular DUB by an activity-based probe, that we have a collection of active compounds in our library, which is really encouraging. I just want to focus in, though, on the compounds I'm really most excited about, which are those that we've now verified as selective through repeat of this screen and dose response. And specifically in this library of 200 compounds, we identified about 25 DUB inhibitors, 25 selective DUB inhibitors with a nice structural diversity that are targeting about 8 or 9 different DUBs. We've now been able to, for a number of these, move these through our pipeline of characterization assays and then start to, investigate some DUB biology. And so this is very much still a work in progress, but, we're very encouraged that we are going to be able to really start to accelerate progress, at least on a subset of DUBs by, using this approach and now having an expanded set of, hits to really develop into highly optimized inhibitors. And so just to briefly wrap up, we've now been able to establish a platform really for developing small molecule inhibitors of DUBs. It's been a lot of work, but we're really excited, to see where the field can go. Still lots of, opportunity for improvements in structural biology and more, design insight. But, definitely, like I said, very excited to, be where we're at. And, we do all of this work very collaboratively. So I'm very grateful, not only to the people in my lab, of course, as well as collaborators, both within Dana-Farber and external. And so with that, I will pass it on to the next speaker. Thank you, everyone, for your attention.
Thanks, Sarah. And our next speaker in this session is Dirk Bussière, who is the Senior Director of the Molecular Pharmacology Department at Eli Lilly. Before joining Eli Lilly, Dirk was at Novartis at their biomedical research, and I believe he will be telling us a little bit about the research he has done at NIBR. So take it away, Dirk.
Yes, can everybody hear me?
Yes.
Okay, great. I am indeed presenting work that I did at NIBR before joining Eli Lilly, and today I will be discussing our work on figuring out the biophysical basis of a molecular glue, which was before that thought to be an enzyme inhibitor. And I'll give you a bit of background so that makes a bit more sense. There we go. Inducilam was discovered in a phenotypic screen in the 90s and underwent two routes of optimization. One route of optimization shown here led to ESI 7070, which became known as Inducilam, a compound that worked primarily on solid tumors. Another route of optimization led to ESI 7010, which worked primarily on soft tumors or blood-borne tumors. Interestingly enough, this was the time of the early transcriptional profiling. The two compounds had very different transcriptional profiles. Now, for many years, you can find literally 20 years of literature that state that Inducilam and the related sulfonamides are carbonic anhydrase inhibitors. And indeed, they do inhibit carbonic anhydrase with nanomolar potency. However, now we are capable of doing lethality studies. In other words, taking cells that have point mutations induced in them through polymerase mismatch repair aberrations, and are able to ask what mutations lead to cells being able to survive a challenge with a compound. And if you do this with the sulfonamides related to Inducilam, such as Tesusalam CQS and Inducilam itself, you find that the mutations that allow colon carcinoma cells to survive challenge with these compounds are not in carbonic anhydrase at all. Instead, they segregate to a class of RNA splicing factors homologous to or identified with RBM39, and they tend to segregate within an RNA recognition motif, RRM2. The mutations all map to the face of one helix of this RNA binding motif. Now, Deepak Najarwan's group did this incredible study and showed that the efficacy of Inducilam does not rely at all on its inhibition of carbonic anhydrase, but instead relies on its ability to act as a molecular glue and targeting a certain class of these splicing factors. And this is presented in the science paper listed here, which I would highly encourage everybody to read. Now, he did a pull down to see what ligase is involved in this. And what he did is flag tagged RBM39 and showed in the absence of Inducilam that you tend to pull down primarily just RBM39, as you would imagine. But in the presence of Inducilam at micromolar levels, you pull down the ligation machinery as well as a very specific E3 ligase, DCAF15. DDB1 is a scaffolding factor for the E3 ligation machinery, and DDA1 was a protein of unknown function, but was associated with E3 ligases. So when we undertook the study, we were asking ourselves, how does Inducilam work? And what is DCAF15? How does the E3 ligase recognize RBM39? And there are three possibilities here. First, you could have an allosteric DCAF15 binder. So Inducilam could bind to DCAF15 or DDB1 in such a way that it modifies the behavior of DCAF15 to bind and recognize RBM39. Alternatively, Inducilam could act as a true molecular glue sitting between DCAF15 and RBM39, or it could act as more of a protac with one portion of it binding to DCAF15 discretely and another portion of it binding to RBM39. And at the time, we did not know which of these would be the actual mechanism. My old lab used to routinely make hundreds of constructs of proteins, but that was not necessary in this case. What we found was that full-length DDB1, full-length DCAF15, and full-length DDA1 expressed and was quite stable. What we did find was that the DCAF15 DDB1 complex required DDA1 to be stable, otherwise it would tend to destabilize. The target RBM39 was easily produced in E. coli. A biophysical analysis through ITC showed that Inducilam did indeed function as a molecular glue. And what you see here is that in ITC, Inducilam has a very low affinity for DCAF15 on the range of 100 micromolar. It literally has no affinity for RBM39, as shown in the second plot. But the complex at DCAF15, DDB1, DDA1, and RBM39, Inducilam binds to at the range of 190 nanomolar. So this is a common aspect of a glue in that it targets its complex with high affinity, but the components of the complex it tends to target with very low affinity. The structure solution was fairly challenging. We put the complex into crystallization trays, came up with these beautiful hexagonal crystals that turned out to have 83% solvent content. Diffraction vanished within a few minutes of exposure to the X-ray beam, as is often common with high solvent crystals. We managed to stitch together a 6 angstrom data set and did a quick molecular replacement and found that the culprit of this was in the DDB1 portion of the protein, which is a trilobed protein. The middle lobe of the protein, shown with the large red X, was inducing a packing that led to a large honeycomb of solvent. Luckily, we had a construct that had this domain deleted. We put that into trays and got very, very nicely diffracting crystals to 2.3 angstroms, which were readily able to be solved. So this is the structure of the complex. And in blue are the 2 lobes of the DDB1 complex, pardon me, of the DDB1 protein. In green, you see DCAF15. In yellow, you see DDA1, which essentially holds DCAF15 and DDB1 together. And in purple, you see the target protein, RBM39. And sitting in the cleft, shown here, is the compound itself. So just to go through the molecular architecture a bit. When Al had asked me to speak, he was interested in WD proteins. DCAF15 is kind of a WD protein. It has some similarities to them, but it's a unique fold. I suspect many of the E3 ligases will have unique folds depending on their functions. We also addressed the structure by cryo-EM. Here you can see the class averages. We had about an equal mixture of dissociated proteins as we did complex. So we had to select the image classes very, very carefully. You can see them here lined in red. We were able to solve the structure by EM to about 3.3 angstroms. The structures are equivalent. Major differences in the EM structure. The compound is shown at a slightly higher energy, of course, because of the fact that crystal structures tend to demand lower energy poses. The RMSD of the two structures is 1.2 angstroms, essentially they're equivalent. So a little bit about the molecular architecture. DCAF15 docks into DDB1 using a docking helix, which is fairly common for members of the class. There is no alphabet to the docking helix. Each E3 ligase seems to engage DDB1 in a unique fashion at a unique angle. And that's shown in green right here. DDA1 literally holds DCAF15 shown in green and DDB1 shown in blue together. It makes interactions with one lobe of DDB1, snakes along the length of the protein, and then bridges over to DCAF15 where it makes interactions, many of which are mediated by waters. And now we get to the mechanism of Indusulum. What happens is, as you would suspect, the helix where the mutations segregate to is actually what is recognized by DCAF15. And Indusulum lies between the RBM39 helix and the ligase itself, shown here. So the E3 ligase embraces its target with Indusulum bridging the interactions between the two. How Indusulum functions is it supplies the lacking complementarity between DCAF15 and RBM39, the target. So you can see it here. The indole ring sits into a hydrophobic pocket, and then each of the sulfamides make discrete interactions either with the E3 ligase or RBM39. And most interestingly, many of these interactions are not direct. They're actually bridged by waters. So Indusulum acts as ensuring surface complementarity between the two proteins. Now, how does this work in practice? This is a mock-up of a E3 ligase with the entire ligation machinery attached. You can see the E3 ligase here, DCAF15, RBM39 in purple, the glue shown as a van der Waals surface, and DDA1 here holding everything together. The missing lobe that we deleted for the crystallography, the central lobe of the DDB1 protein is here. It essentially acts as a swivel. It's highly flexible. And the rest of the ligation machinery attaches to that with the ubiquitin here ready to be attached. And this essentially this scorpion tail scans the surface of the scans the surface of the E3 ligase, looking for a lysine to which to attack the ubiquitin. So this modular design allows this system to recognize multiple proteins and tag them for degradation. Once they've had 2 to 3 ubiquitins attached to them, they are targeted by the proteasome and digested. So in conclusion, DCAF15 is a novel E3 ligase. It's distinct from other previously identified E3 ligases. Indusulum is a molecular glue that sits between the E3 ligase and the neodagron, which is RBM39, as well as a related protein, RBM23. It's probably more challenging to target in a PROTAC approach than cereblon because of the geometries involved and because of the relative shapes of the proteins, but I think it's still possible. Something to note is that the neodagron, that is what is recognized by the E3 ligase in the presence of Indusulum, and the native dagron are two very different things. We don't actually know the native dagron for DCAF15, although I suspect it is a helix, but what sequence it has, I would have no idea. And with that, I'd like to thank my colleagues at Novartis for their help and their participation in this project, as well as their contributions. And I would like to thank you for the invitation to speak, and I'd be happy to answer any questions.
All right, thank you, Dirk, and thank you to all the speakers in this session. We have a couple of questions that have come in, and perhaps we can start with a question related to ABP profiling of DUBs. And that's, do UB row selectivity and ABP profiles correlate across all the DUB family members?
Yeah, Yeah, so, that's definitely a question that we've been excited to address now that we kind of have, both platforms and enough compounds to, really put through and see some general trends. And the answer is that in most cases, we see pretty good correlation, but as you are probably not going to be surprised to hear, there are exceptions. Some of the exceptions we understand quite well. For example, USP7 catalytic domain is often used for biochemistry. We had a really interesting scaffold. It had no activity in ABPP. So once we tracked it down, it wasn't inhibiting the full length enzyme. That one makes sense. There are some other examples, that we haven't been able to really, track down yet to know if we're seeing a false positive in one assay or the other, or if there's relevant, differences in constructs that, can account for the disparity. But by and large, we see pretty good agreement, which we have, found comforting.
Yep. So there are a couple of questions about the Indusylum DK15 RBM39 story. And they all come down to the question of whether in some ways HTS is the only way to find glues. Can these interactions be somehow predicted? And then whether the RBM39 is actually an endogenous substrate for the Cal4 ligase. Like, was there any way to predict that you will find what you discovered?
So my suspicion is that what glues do is modulate nascent affinity between E3 ligases and some not native target, but something that could have been if things were slightly different. So I'm sure that if you put DK15 and RBM39 together and look very carefully, say by bioNMR, you're going to see an affinity. It's not lacking complementarity for each other. But in a biological sense, DK15 does not recognize RBM39. This is pure happenstance. My other suspicion is that many other compounds work at least partially as molecular glues, and we just don't know it. So there have been, recently there was a JMed Chem paper showing a compound that had molecular glue-like properties. They made a very small change in the scaffold and it went back to being an active site inhibitor. So change of 3 atoms changed everything. Is there a way to design it by modeling? If I think you were able to take an E3 ligase that had some affinity for a potential target, and were able to use biophysical methods to get some idea of what that encounter complex looked like. In other words, it would be a very transient complex. If you were able to do that, say through NMR or mass spec coupling, you might have a chance of designing a molecular glue. Otherwise, I think your bet is much better to start with a E3 ligase and a target that have some affinity for one another and do proximity assays to figure out, actually screen to see what induces a complex formation between the two.
So a question for Peter, and I'm curious about this as well, is there a way for somebody to nominate an understudied target for some of the IDG efforts? Like, is there a way for somebody frustrated with how much is known or how many tools are available for their favorite understudied target to come to IDG and nominate something?
Yes, it's not the way that IDG was set up, but you should absolutely email me or the PIs at Gary Johnson, for example, or Sean Gomez. I can send those around. We can post them on the website because we'd be happy to hear ideas from people. The real problem, when you take the least studied piece of the kinome, is you may be looking at enzymes that are not expressed in any accessible cell type, and or you may have no functional information. So I think there is a mechanism in the US that anyone, in fact, could participate in a so-called RO3 mechanism, which allows people to actually get up to $200,000 in funding to bring a target of interest for kinases or GPCRs or ion channels, and actually then work with the consortium on a protein of interest. But at a lower level of commitment, absolutely email us and we'd be happy to talk to you about it, and certainly to provide any tools that have been developed.
Well, thank you. I'm very sensitive that we are a little bit over time. So I'm going to take this opportunity to thank all the speakers that have shared with us today. To remind you all that these webinars have been recorded and they will be posted on Target 2035 website by the end of this week. And to remind you to tune back in tomorrow for another session that will be discussing some of the emerging technologies that will be important to bring on board as we move forward with Target 2035 efforts. So thank you all again today. Thank you for excellent talks and for excellent questions. And have a wonderful rest of your day.
Thank you all.
Bye.
Bye.